To continue with neural processing and in the retina and beyond, we're going to spend a little bit more time talking about lateral inhibition. And I'm going to come back to, so we just went through how they were measuring and that they were measuring from one of these axons that are leaving the eye. If we look at a circuit like this, and if you can, spin that around in your brain, right? So if we're looking at the little dot of light in the center, just hitting 4, B's firing at a rate of 1. If we hit 3 through 5, and again, this could be in a circle, not just in a line, get it into another dimension there, and we're going to increase the firing rate of B. But then as we get longer and we start to hit, or like a bigger circle, we start to hit 1 through 7, because they are communicating some inhibition through bipolar cell A and bipolar cell C. They're communicating some amount of inhibition, so we're going to slow down the firing rate now of that particular bipolar cell B. I'm going to go through a couple of quick proofs that you never need to do, but I think they're really helpful. I will also say that for every one of these examples, there is a counterexample, because that's what scientists do. We try to find the answers to things, and then somebody else says, well, wait a minute, you don't explain, you might explain X, but you don't explain Y. That's kind of what we do. So if you remember when we talked about measuring sensation and perception, and I talked about the phenomenological report, I put up this, which is called the Hermann grid. The Hermann grid, what you'll notice is the kind of tic-tac-toe board and the 9 dark squares. But then what you'll also see is, if you're looking at, let's say, the intersection to the top right, those other intersections that are in your periphery, they have what's called a gray ghost. It's a little bit darker than the white corridors. We're going to explain why that occurs using lateral inhibition. So if we look at what's happening at the intersection, and so we're looking at bipolar cell, let's say A, or retinal ganglion cell A, that initial response, and if we're going to call the white space 100 and the black space 20, so this comparison of light, we're at 100. So the response of A, its initial response is going to be 100. But then we're also getting some amount of lateral inhibition, and the estimate here is about 10%. If I'm sending 100 units of activation to my bipolar cell, I'm also sending 10 units of inhibition laterally to those bipolar cells around me. And so if we look at the neurons, there are the receptors that are surrounding A. If we look at B, and it's in the white, so it's getting 100 units of activation, we're going to subtract out that 10, right, that 10%. C, we're subtracting out another 10. D, we're subtracting out another 10. And E, we're subtracting out another 10. And so by the end of all of this, and we subtract that 40, 100 minus 40 is 60. So A's final response, final answer, family feud, 60. If we compare this to what's happening in the corridors, so now if we look at D, its initial response is also 100 because it's in the white space, but it's surrounded by some white space, but surrounded by more black space. So if we look at what's happening with the inhibition that's coming to it, A is getting 100 units of activation, sending along 10% as inhibition to bipolar cell D, so minus 10. F is in one of those black spaces, so it's getting 20. 10% of that is 2, so there's another minus 2. G is in a white space, it's going to send 10 units of inhibition. H is in a black space, it's going to send 2 units of inhibition. So now I'm subtracting out 24 from 100, and D's final response, final answer, family feud, 76. So what we see here, right, is it's a relative amount of light that we are seeing. And the relative amount of light, given the fact that we have this lateral inhibition, means that A appears to be slightly darker than D or those other areas of the corridors, and those appear to be white, so we get this little gray ghost there. And this is happening where I'm in the periphery, because that's where we're having more of that convergence. And some of that convergence, that complex circuit, is more complex because it includes lateral inhibition. The picture in the book takes away the numbers, and if you don't like the numbers, you really can just ignore me. You don't have to memorize, you don't have to remember any kind of mathematics. Some people it helps a lot. Some people it's just annoying, but if you let it kind of wash over you, that works. This is another way to picture that 3 through 5 are sending excitatory information to B, and this is a lot like the circuit we looked at before, right? Except now I'm showing you the circle that we spin that around, because this is a center surround receptive field, again, on the retina, and the space of the retina of the eye. And so now 1 and 2 are getting excitatory information to A, but that's sending inhibitory. It has an inhibitory synapse with B. 6 and 7 sending excitatory information to C, but that has an inhibitory synapse with B. And again, you can do this thing of looking at one particular intersection and realizing that that's your foveal cones that have a more linear circuit, so we don't see that same gray ghost. What's happening in the periphery where we have these center surround receptive fields that are overlaid on top of each other, basically, right? Because they're coming from all of these areas next to each other on the eye. Then we're going to see those gray ghosts at the intersections, because there's more light that sends more lateral inhibition. And here's a thought question in green for you to pause and think about: why would we want lateral inhibition as part of our sensory system? I'm going to tell you that we don't want to see absolute amounts of light. What we want to see are relative amounts of light. That helps us to see the changes when something changes in our environment. That's what relative amounts of light allow us to see. Whereas absolute amounts of light are going to be this kind of light upon light upon light. And we're not going to be accentuating those changes in the same kind of way as when we have this lateral inhibition that allows us to see those changes. So, and like I said, you could go to an alien planet or another universe. This is the way the sensory systems are going to have to work. I know I said this early on now, and I'm coming back to it now. It's not just about, if we're all just yelling, you don't hear a specific voice. What you want to hear are the voices where there are changes. And so if I'm trying to get attention from my family or from my mother or father, I'm going to not just yell as loud as my siblings, right? I'm going to ask them to shush so that I can be heard, so that I'm louder. And that's what we're kind of doing with a sensory system, is where we want to put our attention is where there are changes, so that I can see where the table ends, or where the chair ends or begins. But, so that's a lot of explanation for lateral inhibition. I've actually cut out a few things to try to shorten it, because I usually pull off onto the projector and onto the whiteboard and other ways to draw things out, and I can't do that over on the PowerPoints as easily. So I will apologize if you don't really get it. It's okay. This is what you need to know, is this sentence, and this will be a short answer matching, not short, a matching fill in the blank. You will fill in the blank. This will basically be just about almost exactly what that matching statement says, where you're filling in the words lateral inhibition. Lateral inhibition refers to the fact that an excited neuron may reduce the activity of its neighbors. This means that if a single neuron is activated, it responds more strongly than if its neighbors are also stimulated, because they are often sending inhibition to neurons along the same path. And lateral inhibition increases our ability to see contrasts and changes. One of the ways I've tried many ways to help people to kind of understand this. Although, again, go back to the last slide. Remember, these PowerPoints are out there for you, and you can just take that slide and write it out for yourself and say, this is what I really need to know. But understanding it on a deeper level is really helpful for understanding how the sensory systems work. So another way to think about this is we'll come back to this when we talk about touch. And if you think about touching yourself on the arm, so I want you to actually do this. People don't do this in class, but you're at home. There's nothing embarrassing, right, about touching yourself on the arm. Touch yourself on the arm with the tip of your finger. And think about the fact that you can tell where that touch is occurring, where that pressure kind of starts and stops. Now, in that same area, take the palm of your hand and touch the same area. Now, the palm of your hand, think about where your finger was. You can barely tell that something's there. You can, but it's not as noticeable. What you notice now, right, is where that touch starts and stops. And that's because of these center surround receptive fields that we're really accentuating where the change occurs. And that's what we want to do. I'm going to just read this as it is. I usually go through this just as it is in class to just put us where we are. How did we get here? Light reflected from an object into the eye. Light focused to form an image of that object on the retina. Light in a pattern that illuminated some receptors intensely and other receptors more dimly is absorbed by those visual pigment molecules that are packed in the outer segments of the rods and the cones. Chemical reactions in the outer segments of the photoreceptors, they transduce the light into electrical signals. Transduction. As these electrical signals travel through the retina, they interact, they excite, they inhibit, and so the electrical signals reach those retinal ganglion cells, which, because of this processing, lateral inhibition, that they're interacting and exciting and inhibiting, they have center surround receptive fields. After being processed by the retina, these signals are sent out of the back of the eye in the optic nerve, and the optic nerve is that cable of nerve fibers or axons of the retinal ganglion cells. So we finally get this information to the brain. Woohoo! The first place we're going to synapse, we're actually going to take that information, we're going to synapse in different places. Different neurons are going to synapse in different places. They're going to synapse in the lateral geniculate nucleus, which is up there, the LGN, the lateral geniculate nucleus of the thalamus. So for all of our sensory systems, we're going to go through some thalamic nuclei. Thalamic from the thalamus, nuclei, because it is a bilateral structure, it's in both sides of the brain. So the majority of the signals for vision go through the optic nerve and reach the lateral geniculate nucleus of the thalamus. We also have the superior colliculus. About 10% of the signals go to the superior colliculus. This is going to take in other information, but then it's going to do this. It's going to take in all this information to build these spatial maps and to tell us where do we want to move our eyes. And if you remember early on when I talked about creating a perceiving machine, one of the huge difficulties is getting just the robots, the cars, giving them enough cameras to make up for the fact that we really don't understand how the superior colliculus works. They've done lots and lots of research on all kinds of animals to try and figure out what's going on with the superior colliculus, but it's controlling our eye movements. If you're looking closely at that brain over to the image to the right, you'll notice that it looks like the lateral geniculate nucleus is there pointed. That's actually in the forebrain, and it looks like the superior colliculus, or because it's bilateral, the superior colliculi is the plural. It looks like it's behind, but it's actually more like beneath. It's the superior colliculi are in the midbrain. So we're taking the majority of those signals to the lateral geniculate nucleus, and then the lateral geniculate nucleus passes that information on in these striations, which is what those little purple wavy things are, in these striations going back to the primary visual cortex. Sometimes and often called the visual cortex, sometimes and often called V1 for visual cortex 1, primary, but then sometimes called the striate cortex as well, just so you know if you're out there and they're talking about the striate cortex. It has this striped appearance, which is what striate means, that it's striped. And so the visual cortex is receiving information from the lateral geniculate nucleus, the LGN, and it's going to pass that information along two pathways, one going ventrally through the temporal lobes and one going dorsally through the parietal lobes, both coming from that visual cortex in the occipital lobe and moving forward through the brain. If we look at these primary visual pathways and what's happening when I'm looking straight ahead of what's hitting the retina and then how this goes back to the brain, we see a contralaterality that is more complicated than a lot of the other contralaterality that we have in the brain, which contralaterality means contra going across, and the lateral is the sides. So it's going to the other side. It's crossing to the other side of the brain. But in the case of vision, it is the left part or the right part of the visual field that's actually crossing to the other side. So if I'm fixating straight ahead, you can see that what that means is that that tree in the picture, the tree is on the left and then the house is on the right when I'm looking straight ahead. And what that means as far as, because we have that inverted flipped image on the back of the retina, the house is hitting the left side of each retina, the tree is hitting the right side of each retina, and you can see this being represented in the brain, that we're keeping this upside down, backwards, this flipped image in as we get to all the way to the visual cortex. So the left half of each eye receives the images from the right visual field. The left side of the left eye that is closest to the temples is going to go back ipsilateral. It goes along the same side. The left side of the right eye that is closest to the nose, that has to cross over. And we're going to see this crossover. Those crossovers occur at the optic chiasm. That's where we have this crossing over of those inner parts of the eyes. And just so I do it in the other direction as well. So when we're looking at straight ahead, what's in the left visual field, it's hitting the right half of each retina. And so the right half of the right retina is going to go back ipsilateral. That's the part that's closest to the ears or the temples. And then the right part of the left retina that's closest to the nose, it's going to have to cross over to the contralateral side. That information is going to get sorted together in this miracle of embryological development that gets sorted together in the lateral geniculate nucleus, and then we pass that back along. The lateral geniculate nucleus is doing a number of things. It is organizing information. It's controlling the flow of information. I often talk about these thalamic nuclei for sensory information as being like the librarian of the brain. So summarizing, filtering, and organizing that information so that we can find the information that we want, right? The books that we want. That's what librarians do. It gets input from many places. So from the retina, other nuclei in the thalamus, from the cortex, we see these white matter tracts coming back when we talk about top-down processing. We see white matter tracts communicating from the frontal lobes to the cortex, and from the cortex back to the thalamus, to the lateral geniculate nucleus. It receives input from itself, so from within itself, as well as from the brainstem. I often do this little clap, which gets really attenuated, I've noticed in the videos, but so if I wake somebody up in class or I see my dogs and their ears stick up as they're orienting to this sound, that information is also being sent to the lateral geniculate nucleus, because some information is more important than other information. And so that also influences what gets sent on to the primary visual cortex. So the output from the lateral geniculate nucleus is primarily to the cortex, actually to V1, to the primary visual cortex. So we see this 4-to-1 ratio of afferents, or arriving fibers, to efferents, or exiting fibers. It's getting input from many, many places. It's getting these 4-to-1, so 4 times as much incoming information as outgoing information. And it is doing this summarizing, right, so that we can find the information that's going to be important for us. Signals arriving in the lateral geniculate nucleus are sorted and organized based on a number of things. One, the eye that they came from. So if I'm getting my left half of my left retina and my left half of my right retina filtered together, right, then those are going to be sorted and sifted kind of together, but they are in their specific layers. So they're organized based on the eye they came in from, the receptors that generated them, whether those be rods or cones or short wavelength cones, and then also the type of environmental information that they represent.